At Collaborations Pharmaceuticals, we use real drug discovery intelligence alongside artificial intelligence to develop clinical candidates for rare, neglected, and unmet therapeutic needs. Our core expertise extends to high-quality data curation and machine learning model building, which we offer as a service. We can apply our technologies to drug discovery and ADME tox predictions. We can also conduct in vitro testing in our own labs and coordinate in vivo testing through our collaborator network. We have utilized drug repurposing to get us quickly to promising molecules, as we have demonstrated for Ebola, Chagas, and Alzheimer's disease. And we can apply these approaches to virtually any disease. We are unique in that we publish our own results in peer-reviewed journals and share our data models, unlike other artificial intelligence companies. Please get in touch to learn more about how we can help you at collaborationspharma.com.
Hello, I'm Sean Eakins, the CEO and founder of Collaborations Pharmaceuticals. Thank you for joining us today to learn about our work on endocrine disruption pathway modeling and how we at Collaborations Pharmaceuticals can help you with your projects. We're very conscious of your time, and we'd like to get through this as quickly as possible so that you can get back to your important work. But we're going to show you how we are committed to using computational approaches in this short time, both for toxicology applications for pharmaceutical, agrochemical, and consumer product uses. We really value your attention, and we'll definitely be taking questions, and we'll try to keep this to 25 to 30 minutes as well. So just a few public service announcements before we begin. We'll be taking your questions, and there's a little icon on the top of the screen that you can see highlighted here. And so you can write your questions at any time, and we'll address them at the end of the presentation. There's also an attendee chat button to the left of the screen, and you can send us messages there as well. And if you want to send us private messages, you can do that too. All non-presenters are muted, so you won't be able to talk to us, but you will be able to write the messages. And as I said, we'll answer them at the end of the seminar. So just a few introductions before we begin. We're going to talk about Assay Central for Endocrine Disruption. And so Kim is going to give a presentation on this today. And then at the end of that presentation, we'll talk about the services that we offer. And then we'll have questions and answers. So that's the plan of today. So just to begin, I'd like to give you a little introduction to those of you that haven't come across our company, Collaborations Pharmaceuticals, before. I founded the company in 2015, really to build upon my 24 plus years of experience in drug discovery, machine learning, and computational toxicology research. We're currently located at NC State University and in incubator space. We have been able to build the company in these few years primarily through grant funding from DoD, NIH, as well as fee-for-service work. And we've been able to fund this to the tune of about $7.6 million without any angel or VC funding. We have numerous orphan drug designations and rare and neglected disease designations as well. There's several patents. This slide just gives you an idea of some of the institutions that have funded our work, and in particular, the NIGMS and NIEHS have funded the work on our machine learning modeling approaches that are highlighted today. We've also been fortunate to receive matching grant funding from the North Carolina Department of Commerce and the Department of Defense as well. But more on those at some later time. So our company goal really is to use our machine learning expertise, both in data curation and machine learning, to identify and develop clinical candidates. And along the way, we've been using these technologies also for computational ADME and toxicology property prediction. We basically follow the format shown on the right of the slide, where we can curate data, build models, generate predictions, and then obviously perform experiments in vitro. And in some cases, we work with collaborators or CROs to perform in vivo experiments as well. Our key focus, though, is really on this hit-to-lead candidate generation, or small molecule machine learning primarily. And our particular focus is in really challenging ADME tox problems. As you'll see today, we've focused primarily on endocrine disruption, but we also have numerous models focused on toxicology, as well as biodegradation and bioaccumulation as well. And we love to work on new challenging data sets. More on that later. As a small company, we have an extensive pipeline working on a number of infectious and rare and tropical diseases. And most of these have been supported in some way by machine learning, either for identification of the molecules or for optimization or toxicology prediction. And this slide just gives you an idea of some of the vast array of projects that we work on within the company. So without further ado, I'd like to introduce Kimberly Zorn, who's a senior scientist here at Collaborations Pharmaceuticals. She was one of the first employees at the company and joined in February 2017. And pretty much since that time, she's been working on endocrine disruption as well as a number of other projects. And she's been able to present her work at really prestigious conferences at CTAC, ACS, and ICRS. And she's published numerous papers. Actually, it's over 17 now. The latest was published just today on aromatase, but I'm sure she's going to tell you about that. So without further ado, I'd like to pass it over to Kim. Thank you, Kim.
Thank you, Sean. So first, we're going to start with just the very, very basics. What is endocrine disruption? Endocrine disruption is the alteration of hormone levels by chemicals in the environment or consumer products, for example. And this imbalance can affect normal skeletal and sexual development and reproduction, as well as causing cancers. Key targets of interest for research include the signaling pathways induced by the estrogen and androgen receptors, or ER and AR respectively, as well as the inhibition of the enzyme aromatase that converts androgens to estrogens. There have been regulatory pushes for rapid and cost-effective methods to anticipate endocrine disrupting effects from chemical exposure, such as the Endocrine Disruption Screening Program from the United States Environmental Protection Agency. But chemical backlogs in testing and evaluation are already immense, and animal work is costly and time-consuming. So, with the added pressures from animal rights groups, computational methods offer a solution to prioritize and triage compounds for testing. In that vein, the EPA used over 1,800 chemicals with in vitro data from ToxCast and Tox21 assays to create pathway models of ER agonism and antagonism, shown here in blue and red respectively. The component assays of these pathways cover a variety of biological processes like receptor binding and transcription to offer a more complete picture of endocrine disruption via ER. The model itself assigns an area under the curve, or AUC score, to each chemical that is binned so that compounds are classified as either active, inactive, or inconclusive. These AUC scores are generated from the experimental AC50 data for each chemical. The data reported in each component assay is standardized and then combined to yield this score, so that the structure of the molecule itself is irrelevant when calculating this value. These pathway models were validated with multiple test sets of in vitro and in vivo classified chemicals. Furthermore, there was also a consortium effort to create consensus computational model called SURAP, with these classifications available for download from the EPA CompTox dashboard. Later on, in 2017, the EPA applied similar methods to generate AR agonist and antagonist pathways, shown here, as well as the COMPARA consensus model published earlier this year. Again, these models were validated with both in vitro and in vivo chemicals, evaluating literature and confirmatory assays, as well as establishing confidence scores. So the question we asked ourselves is, can we anticipate endocrine disrupting effects with Bayesian machine learning models generated in our proprietary software, Assay Central? And how does this compare to the work already done by others at the EPA and elsewhere? I will mention that at the time of preparing this work, SURAP and COMPARA were not capable of prospective prediction. However, this is a relevant evaluation of the methods using similar datasets. So before I get too far ahead of myself, let's briefly cover what I mean when I say Assay Central. Simply put, this is a technology that is a framework for curating structure-activity datasets and building machine learning models. It applies a Bayesian algorithm and utilizes extended connectivity fingerprints, or ECFP6 descriptors, to conduct five-fold cross-validation. This generates internal performance metric summaries, such as the one shown here at the left. For classification models such as these, an activity threshold has to be defined. But this software is also able to calculate a threshold to optimize training performance when one is not manually specified. Assay Central also allows the user to generate predictions. This is done by first calculating the ratio of a given fingerprint present in active and inactive compounds within the training data, deemed a fingerprint's contribution. When presented with a prospective molecule, the summation of the fingerprint contributions present in the external molecule generates the probability-like prediction score, and we consider an active score as one greater than or equal to 0.5. In 2018, we published on utilizing 8 datasets from multiple sources to compare the training and external validation performance of Assay Central's algorithm and descriptor to different combinations of 5 other machine learning algorithms and descriptors. Overall, we observed a similar performance in training and test set prediction metrics and concluded that classical machine learning algorithms, such as the one used in Assay Central, are sufficient for datasets such as these. What I am about to present is our continued efforts that have evolved from that preliminary study. Herein, we have used Assay Central to create groups of machine learning models from the same component assay datasets in the EPA's ER and AR pathway models. We compared different data types, including the AUC output scores generated from the pathway models, as well as the raw AC50 values and binary hit calls available from the ToxCast download page. Furthermore, we have also included burst flag hit calls, which classify a compound as active only if the reported AC50 is below the measured cytotoxicity. For the AUC models, we used the EPA's published threshold of 0.1 for an active designation, but we opted for an automatically calculated threshold for our continuous AC50 datasets. Assay Central model groups were evaluated with the same test sets as the EPA did, but herein we generated true external predictions by first removing these chemicals from the original training datasets. Prediction scores from component models within a group were combined in a majority rule fashion, so if more than half of the component models in a group could predict the chemical as active, then the overall designation was an active designation. The AUC models were the exception to this as they were standalone datasets. When evaluating the ToxCast pathway model prediction accuracies, know that only the raw AUC score was considered here. We did not evaluate confirmatory assays or literature data as was done for the AR pathway model validation with in vivo chemicals, for example. Know that both of these studies are published, as Sean already stated, and I am only presenting a sample of what was done. There are far more details available in these articles, and I highly encourage you to go out and read them. I'm going to start by discussing the ER agonist pathway, made up of 16 component assays. 3 test sets taken from previous EPA publications were used in our evaluation. These include a set of in vitro and in vivo reference chemicals, as well as another set of chemicals with uterotrophic calls from guideline-like assays that didn't quite meet the standard for reference chemicals. So here we have our first graph of prediction results for the in vitro test set. On the x-axis, we have our various models. The consensus and pathway models from the EPA are shown in dashed bars, just to differentiate them from the Assay Central machine learning model groups in solid colors. On the y-axis is just a standard frequency count. Gray indicates the total number of correct predictions overall, with navy and red bars representing the number of active and inactive predictions made by a model or group, with the number of correctly predicted active and inactive chemicals in light blue and gold. Here, you can see that generally all of the models performed similarly. While the EPA ToxCast pathway model was best in that it produced no false positives and only 2 false negatives, the Assay Central group utilizing binary hit call data came very close and actually yielded the same accurate prediction total as the pathway model. Next, we have the in vivo reference test set. In this case, the same binary hit call group that performed well on the last slide was actually the worst for this test set, with several false positives and few true negatives, specifically. Despite this, all Assay Central model groups were able to predict 29 active compounds correctly, with only 1 to 2 false negatives, which is similar to both of the EPA models. Here, the burst flag hit call model group was able to accurately identify the most chemicals overall, as well as the most inactive chemicals, slightly surpassing the syrup model. Finally, we have a test set of 49 uterotrophic calls that were not in the previous in vivo reference set. This was clearly the most difficult for all of our models involved, as demonstrated by the prominence of false positives and negatives across the board. The ToxCast pathway model produced the least false negatives, but the burst flag hit call group produced the least false positives, as well as the most correct overall. However, all assay central model groups are again performing similarly to the EPA models. In summary, the assay central models performed as well as, if not better than, the EPA consensus and ToxCast pathway models across all test sets. The pathway model was best at limiting false predictions overall, but Assay Central performed similarly without using inconclusive scores. The guideline-like test set was the most difficult for all models, though, indicating the very familiar difficulty in translating in vitro activity to in vivo prediction is still elusive. This is not entirely unexpected, as there are inherent variability in vivo studies. There is also room for improvement in that we observed certain problematic chemotypes. For example, phthalates were classified differently in vivo and in vitro reference datasets, so depending on the test set, they were predicted accurately or not. Interestingly, the EPA had similar issues, and these are all really simple chemicals, so additional descriptors like molecular weight might be necessary to improve upon our predictions. Now, we'll go ahead and move on to AR. We'll focus specifically on the antagonist AR pathway, composed of 7 individual assays. We tried something a little different for this study, in that we evaluated if removing a large portion of the training dataset that was determined to be inactive and thus assigned an AC50 value of 1 million micromolar would impact the results. The rationale here was that if the calculated threshold was still applied, removing these inactive chemicals would yield a more useful and realistic activity cutoff and ultimately help out with our prediction accuracy. I'll refer to this specifically as the active-only AC50 datasets. 2 test sets were evaluated herein, again with either in vitro or in vivo classifications. And as a reminder for the upcoming slides, again, only the AUC output from the pathway model was considered for these prediction accuracies. In the published validation of the AR pathway model from the EPA, they further evaluated literature and confirmatory assays that we have not done here. So here we have, again, our familiar graphs for the in vitro AR antagonist test set. As you can see, the EPA ToxCast pathway and Compara models are the clear winners overall here. The ToxCast pathway model actually correctly identified all of the 8 inactive chemicals and almost all of the active chemicals correctly, with only 1 false negative observed, and Compara produced none. It is also apparent here that the datasets attempting to limit false predictions did not work as intended. The correctly predicted active chemicals for either the active-only AC50 group as well as the burst flag hit call group total less than half of the others, and both produced many false negatives in the process. Interestingly, the other assay central model groups appear to be more useful at identifying active chemicals specifically, matching or surpassing the EPA models. And while there are fewer accurate negative predictions, there was only 1 false negative among them. For the in vivo test set, we see more false negatives than false positives immediately. Assay Central hit call and the full AC50 groups were the most accurate overall, performing better than both the EPA pathway and the Compara models. As in the previous slide, the model groups using active AC50 values only and the burst flag hit call datasets did not perform well at predicting in vivo chemicals either. The AUC model was unique here in that the false positives exceed the false negatives, indicating that it might be more ideal for predicting AR inactive chemicals specifically. To summarize, the Assay Central models were superior to the EPA models at predicting in vitro chemicals. Similarly, the in vivo test set was challenging for all models, but 2 of the Assay Central model groups produced more accurate predictions overall, with less false negatives than the EPA models. Now, there still were some false negatives, and this is still an area to revisit in future studies, though. Removing the inactive AC50s did not improve predictions, and generally the burst flag hit call models were largely ineffective for our AR antagonism predictions. Overall, this test set demonstrates that the issues in predicting in vivo results with in vitro data is still elusive again, and the false negatives were common throughout. We have also gone a step further in our efforts to predict endocrine disruption by modeling aromatase inhibition. Again, this enzyme converts androgens to estrogens. Training data was pulled from either ToxCast or Tox21 assays, still evaluating different data types, but this time AC50 models were set to a threshold of 100 or 10 micromolar. And we evaluated these models with multiple test sets again. One was from the Chembl database, and it had percent inhibition data that totaled nearly 700 compounds, where we considered greater than 75% inhibition to be considered active. And this was predicted with both of the AC50 thresholds. The other test set was from a single literature source of IC50 data, where we set the threshold to 10 micromolar and did not utilize the 100 micromolar training model. This is because even at 10 micromolar, the entire data set was almost active, so there was no real need to compare it at the 100 micromolar threshold. To briefly summarize, we found that generally the Tox21 assay was the better predictor of the IC50 data, and the ToxCast assay was better for the percent inhibition test set. There's a couple things that could be going on here, though. This is possibly due to the size and coverage of chemical space of each of the data sets, as we noticed that the ToxCast models always possessed fewer than 70 actives out of their 1,700 total compounds. Additionally, the assay technology likely plays a role here, highlighting the importance of understanding your input data. The ToxCast assay measured cell-free binding to aromatase, while the one from Tox21 was a reporter assay. As they say, the devil is in the details, so there's plenty to be explored here in future studies. Also, similarly to what was done in our 2018 ER paper, we have compared 6 other machine learning algorithms for all of the endocrine disruption datasets I have discussed thus far. However, rather than evaluating different descriptors in addition to the algorithms, we restricted the comparison to only the ECFP6 descriptors that are used in Assay Central. We have previously detailed how we use a rank normalized score to evaluate the 5-fold cross-validation metrics of our various models. Briefly, all the metrics generated by a certain algorithm are range scaled between 0 and 1, and we examine the median of these. An example is shown on the left here for AR. We further refine this to a difference from the top metric, which subtracts the rank normalized score of each algorithm from the highest score overall. An example is shown at the right again for AR. This ultimately allows for a direct comparison of the performance of 2 algorithms while maintaining information from the others. And our big takeaway from each of these studies is that Assay Central is competitive with these other machine learning methods, including more advanced ones like deep learning. But the underlying Bayesian algorithm allows for quick generation of models on a standard desktop computer, and it has shown to be more robust for unbalanced datasets with few actives, which is a common occurrence for our various disease projects. In summary, we developed a suite of machine learning models for endocrine disruption, including those mimicking the ToxCast pathway models from the EPA, with our proprietary software Assay Central. Rather than based on the variable experimental data from each assay, our technology offers the advantage of generating a score based on the chemical structure of your predicted compound. In our evaluation of these models, they performed on par or exceeded the performance of the EPA models when evaluating external predictions of in vitro and in vivo test sets. We plan to apply these models in tandem to the company pipeline shown previously, so as to filter out potential endocrine disruptors from our testing requests. We also plan to build on this study with even more machine learning algorithms like XGBoost, as well as comparing predictions with those models, as only Assay Central was evaluated herein for that specific those test sets. Additionally, since most people really don't want to interpret tens of model predictions, it's highly desirable for us to develop a method of a consensus prediction across these model suites. Finally, we do aim to compile more assays and targets to construct our own steroidogenesis pathway, utilizing all of the work I've discussed. Finally, I would like to give my thanks to the EPA for laying the groundwork and supplying all of this data publicly, as well as, of course, our government funding from the NIH, specifically NIGMS for Assay Central funding and NIEHS for Megatox funding that Sean will discuss in a little bit. And of course, we would like to thank our machine learning gurus, Daniel P. Russo and Alex Clark. And last but not least, I really have to give a big shout out to the SC Johnson team for the countless brainstorming sessions and keeping me sane and help me interpret all of this data. And with that, thank you for your time, and I will turn it back over to Sean.
Thank you very much, Kim. That was fantastic. Wonderful. And yeah, I'd love to reiterate acknowledgements to all the folks involved in this particular project. It's a labor of love. So we're very happy to take any questions you may have. Just as a reminder, please use the audience question button at the top of the screen or the attendee chat. And I'll just give you a few minutes to put your questions in either of those places, and then we'll answer them. And while you're doing that, I thought I would give you a little overview of the types of things that we can offer at Collaborations Pharmaceuticals. Obviously, as you've seen from what Kim's presented, we can definitely do data set curation and machine learning. We have access to multiple algorithms and technologies for building models like deep learning and random forests, etc. We have a lot of expertise with both very small data sets from the ADME side of things, and then also very large libraries of molecules. We've been using a number of different approaches to prioritize compounds for both synthesis and testing. And we also, as mentioned by Kim, have methods for sort of consensus ranking of predictions. And we've used these quite widely to try to bring together target activities as well as off-target activities. Finally, we have our own technologies for calculating synthetic feasibility and retrosynthetic analysis of compounds as well. So if you'd like to design new molecules, we have those capabilities. And as I mentioned at the beginning, we're very keen to accept new challenges, and we'd be happy to help you with your projects. In the process of our work funded by the NIH, we've been able to develop, as Kim showed, Assay Central, which is the core technology for both curation of data and building and validating models. And in the process of this, we've been able to put together collections of models in different areas. And obviously, the endocrine disruption models fall very nicely within our Megatox package, which is a subset of toxicology and molecular property models that we've curated from literature data sets. So, for example, Tox21 data sets from Kemble, etc., and also the literature. Additionally, we've been able to put together a collection of efflux and uptake transporters, a product we've called Megatrans, and we've been working diligently on that, so hopefully we'll have some more publications on that in the very near future. Watch this space. And finally, MegaPredict, which is a compilation of thousands of Kemble models that we've built, and this allows us to come up with scoring of molecules against these thousands of models to try to come up with ideas of off-target effects as well as potential drug repurposing ideas. If you'd like to try Assay Central, we have a website, www.assaycentral.org, where you can try to use these models. We have a number of target-related models as well as off-target models, and a number of molecular properties are in there as well for various ADME properties. And finally, obviously, with the COVID-19, we've been involved in building models and curating data sets for activity against COVID, as well as additional targets relating to coronaviruses. If you'd like to find out more about the Assay Central underlying technologies, we have a number of publications with Alex Clark that were published in 2015. And then over the last few years, Kim has published very widely on applications of Assay Central in a number of areas, as she's shown today on endocrine disruption, as well as other areas of toxicology. Finally, I'd just like to mention, obviously, we've been able to work with a number of companies in the consumer product area, and these represent several client use cases where we've been asked to curate data sets, build models, and in several cases, obviously publish that data and give presentations. We've been also able to work with a client that's a rare disease company, where we were tasked with coming up with ADME models for them and coming up with new molecule designs. And we've had multiple projects with all of these companies to date. And finally, a small animal model CRO, where we've been involved in both predicting activities and in vivo activities for molecules, as well as ADME properties. All of these client use cases really are examples where we've had a close collaboration with the companies involved. So with that, I think Kim and I, we've now given you a bit more time to answer some questions. So we're just going to take a look to see what questions we have. Okay, so we have quite a few here, actually. One question, Kim, is what do we plan on doing next? I think you alluded a little bit to that with the steroidogenesis pathway, but do you want to add a little bit more?
Definitely the world is our oyster for Assay Central. I would say the next thing that we can do is anything we can even think of. Steroidogenesis, certainly there's lots of targets within that pathway. We have lots of ideas, fires going as far as creating our own kind of pathway models for disease targets specifically. And I would say, but keeping in our endocrine disruption sphere, as I said previously, as far as in the direct future, I would say we're really wanting to focus on predictions with these alternative algorithms and see if we've evaluated fivefold cross-validation, but we've seen time and time again that our training statistics can be deceptive once you start evaluating predictions. So that's where I would say I'm most excited about our future endeavors.
Thank you, Kim. We have a question regarding the typical machine learning models require quite large training data sets, but in our case, they appear to be small. The question is this hampering the prediction if just small data sets are available? I guess my take on that is for the endocrine disruption data sets for ER and AR, these are quite large data sets now that are in the public domain. Kim, I can't remember how many thousand molecules, but we're in the thousands, right? These are not by any means small data sets now.
I will absolutely agree with that. Our test sets were kind of smaller than some people would prefer. But I would also say that we do a lot of rare and neglected disease work. So our idea of what is small is pretty different from some other people. Of course, we're limited by just what is available publicly. We specifically went based on what the EPA had done there, and I really trust them on how they are curating these test sets and these reference chemicals. We need more manpower. Absolutely. I would love it if we had thousands and thousands of chemicals to test. But unfortunately, we just don't have that right now, and we'll just have to be patient and wait for more data to come out into the public sphere.
I mean, some of these data sets are definitely up in the low thousands to 7,000s. These are pretty big from our side. We've definitely had experience of modeling smaller data sets. The aromatase ones probably on the more small side. So I think the answer really is that definitely that we have a lot more data that's appearing in the public domain. I think part of the challenge is maybe pulling it together as well. So the curation is really critical. And that's what we spend a lot of time, or Kim spends a lot of time on the data curation side of things. Hopefully that answers your question, Stefan. Actually, yeah, I should say that was a question from Stefan Schultz. So thank you, Stefan. We have a couple of other questions as well. One question on how did we derive the rank normalized score for comparing various machine learning approaches? I don't know if you just want to repeat what you described, Kim.
Yeah, so once again, we kind of take these metrics that are generated by a certain algorithm, fivefold cross-validation. They're range scaled, and so they're set between a hard 0 and 1. And then the median of those scores is actually analyzed, and we're comparing it across these different algorithms. This has been detailed, quite extensively in some previous publications. So if you'd like, I can absolutely point you towards those, as well as Dr. Thomas Lane, who actually does all of that analysis. He can absolutely explain that a lot better than I can, I'm sure.
Thank you, Kim. We also had a question on if we were to obtain a set of compounds from extractable, leachable studies of pharmaceutical containers or medical devices, could you run them and provide a report of the predictions? So that was one question. I think maybe the thing I should explain is that given a structure, we're running these predictions just from the molecular structure. So we don't require any in vitro data ourselves. We're basing the models on the in vitro data that's already been generated. But yes, any molecule structure we could run through these models and come up with predictions for them. So whether those are pharmaceutical compounds, agrochemical, consumer product ingredients, scents, dyes, you name it, we could run them through these various models. And we had a question also about can we add a little bit more about how we can use Assay Central and how a user would select the model as output. So where do we draw the line, I guess, on how the models are selected for the predictions? That's a good question.
So this is where I would say that our company is called Collaborations Pharmaceuticals because collaborations is really important. As far as selecting, what models are going to be most useful to you, that's one of those conversations that we have to all have together. You might say this assay is completely useless. I've seen time and time again that it gives completely variable results and I don't want to include it. Absolutely. We can keep that out of our prediction reports completely. As far as how the user would use this, we can actually bundle up our models and distribute them to you directly so you can play with them on your computer, on your time with your private chemicals. And then you can actually have all of the power. You can say, I want to have the outputs from every single model, and I want to have all of the information at my fingertips. You can say, I read this paper over here that said that this assay is the only one that we need to be concerned about. And you can generate all of that data yourself as well. It's just that when it comes to the interpretation, or if you want to get different styles of predictions, for example, the ranked or remove anything that's present in the model, for example, that's where you'll need our help a little bit more.
Thank you, Kim. We had another question about have we shared any of the predictions with regulatory authorities, and what has been the feedback? And are there thoughts about characterizing predictions with confirmatory in vitro assays? Great questions. So far, we haven't discussed these with regulatory authorities. I think that would be definitely something that's desirable. Hopefully, we've got a few folks that have been watching the presentation today. We'll definitely be sharing this with various groups on the regulatory side. I've definitely reached out to a number of them, both in the US and Europe as well, and made them aware of the presentation. And we'll be making this webinar available online as well afterwards. I think from the point of view of validating the predictions, the in vitro side of things, that's definitely something we have capability to do now at the company. And we've been doing this in a number of drug discovery areas where we've used machine learning models to make predictions. We've obtained the compounds and then tested them for different disease areas. We could absolutely do the same thing. As Kim mentioned, part of the complexity is which assay do you use for the various nuclear receptors, for example. And so having a assay that closely relates to one of the data sets that's used for training, that would be critical to have a sort of apples to apples comparison. So definitely that's something we could do. And obviously these models have applications both from the side of environmental, but also from drug discovery side, because many of these are valid drug discovery targets for cancer. So that's definitely something of interest to us. We also had a question about how much easier the database appears to be to use compared to ToxCast. What are the challenges associated with using AUC for making calls on how similar concentration response curve is to something like 17-beta estradiol? Kim, I don't know if you want to add something on that, because you're obviously at the cutting edge of dealing with the AUC data.
I would say challenges associated with data are probably pretty universal to any type of data. You have to know that it's reliable. You have to know where it's coming from. You have to understand it forwards and backwards, really. I think, the challenge with the AUC scores, using those in our models, is that it is, an all calculated. It's not exactly apples to apples, this was all standardized. Some of these chemicals were used at different concentrations. And, I kind of like the idea of testing that output and using that as input into a model, because I've seen that often throughout the literature of people doing that. Is that a good idea or a bad idea? We saw some advantages and disadvantages to it in our studies. And so I would say that, again, the challenges are pretty universal to any kind of data, that you should just be aware, ultimately, is the biggest advice I can ever give anyone, is just be aware of what's going into your model and how you would be interpreting that.
Great. Thank you, Kim. Great answer. Well, if there are no other questions, we've answered quite a few today. We'd be happy to take any questions you can think of afterwards. You can certainly contact us at our email addresses at the bottom here, either Sean or Kim at collaborationspharma.com. If you'd like to find out more about the company, please go to www.collaborationspharma.com. We have a number of past webinar videos there, and you're welcome to look at those. Our next webinar, hopefully in December, will be on megatox, where I'll be diving into some of the computational ADME models and predictions that we've been doing and publishing recently, in particular on drug-induced liver injury and also on acute, rad-acute toxicity, some of the models that we've published in very recent papers. And certainly we'd be happy to send you any of the preprints for the articles that Kim highlighted today. I'd just like to reiterate our thanks to our collaborators and friends at SCJ and everyone involved in these projects. It's been wonderful to present these 3 and a bit years of work now. And obviously, this has pretty much all landed on Kim's shoulders, and I think she's done a fantastic job. So thank you, Kim, for a wonderful presentation. Thank you all for your attention. And we'll be happy to discuss any of your needs further. Please get in touch. Thank you.